What's up, people? You're welcome again in part 2, Pharmacodynamics. In part first, we saw what is pharmacodynamics, that what drug does to the body, right? And in part first, we covered the mechanism of drug action, right? And the principles of drug action. Now, in this video, the part 2, what we are having is two topics. Action effect sequence is our first topic, and the second topic we are having is transducer mechanisms. Okay, and we are going to study these topics in much detail. Okay, so starting with action effect sequence. Now two terms here are important which do look similar, they sound similar, they look similar but they are not. Okay, so drug action and drug effect. So what is the basic difference between the two? We will see this. Okay, so drug action, it is the initial combination of drug with its receptor resulting in conformational change, or it's just delete this, resulting in its conformational change. Okay, so or its prevention. Fine. So what is drug action? ड्रग एक्शन में ऐसा होता है कि इनिशियल कॉम्बिनेशन, जब ड्रग इनिशियली रिसेप्टर के साथ जाके कंबाइन होती है जिससे रिसेप्टर में कोई कन्फर्मेशनल चेंज आए या फिर उसका रिसेप्टर जो है वो उसका जो एक्शन है वो प्रिवेंट हो सके. ओके, सो दैट इज़ व्हाट initial action, initial combination, that is called the drug action. Now what is the drug effect? It is the ultimate change in the biological function as a consequence of drug action through series of steps called transducer. Now drug effect का ये मतलब हुआ कि ultimate क्या चेंज आया जब ड्रग ने रिसेप्टर के साथ बाइंड करा. वी नो व्हाट रिसेप्टर इज, वी सॉ दिस इन द प्रीवियस वीडियो. इफ यू हैव नॉट aware about it, please go and watch part 1 first. Okay, so ड्रग इफेक्ट में ऐसा हो रहा है कि जो फाइनल जो अल्टीमेट चेंज हो रहा है बायोलॉजिकल फंक्शन में, यानी जो रिस्पॉन्स प्रोड्यूस हो रहा है, इट अकर्स थ्रू अ सीरीज ऑफ स्टेप्स, जिस स्टेप्स को हम बोल रहे ट्रांसड्यूसर मैकेनिज्म्स बोलते हैं, राइट. सो दिस इज व्हाट हैपन्स when the drug combines with the receptor. Okay, so the initial change, which is called drug action, and the final change, which is called the final change or the final response, which is called the drug effect, right. Now if we look at the structure of receptor, now it is having two domains, the ligand binding domain and the effector domain. So as you can see in the figure, we are having two domains here, ligand binding and the effector domain. Okay, and what ligand binding domain does, it is used for recognition. It recognizes the drug molecule, right, and it is spatially and energetically suitable for binding the specific ligand. So what do you mean by ligand? We saw in the previous video that ligand is a molecule, a substrate which is having affinity for the receptor. Just that. Okay, it's very simple. So that is what ligand is, a molecule having affinity to the substrate. So a ligand binding domain is spatially in space and energetically suitable for binding the ligand. So this is the place, this is the area where ligand or जो भी drug है हमारी, it is going to bind, right. So the first domain. Then the second, effector domain, it undergoes functional conformational changes. Okay, so this is what this is used in transduction. What do you mean by transduction? These are the events, step of events due to which or ultimately due to which जो final हमारा change है या final जो response है वो produce होने वाला है. Okay, so effector domain, it is the area where conformational changes takes place due to which the final response is going to be produced, right. So this is what a receptor look like. Refer the figure, and it is having two domains, ligand binding and the effector domain. Okay, now we will see what is Transducer mechanisms. Okay, now what are transducer mechanisms actually? So it is a complex multi-step process, right? It is a multi-step process for amplification of the signal, and it produces functional response. Okay, so we saw that the drug binds to the receptor, right? Now after binding to the receptor, a conformational change occurs. Okay, now these the conformational change it gave rise to a complex multi-step process. उस प्रोसेस में सिग्नल का एम्पलीफिकेशन होता है, उसकी इंटेंसिटी बढ़ाई जाती है. ओके, ताकि हम हमारे पास बायोलॉजिकल रिस्पॉन्स प्रोड्यूस कर सकें हम. ओके, so the types we are having of transducer mechanisms are five. Okay, the first one, G protein coupled receptors. Then the second, ion channel receptors, transmembrane enzyme linked receptors, transmembrane Jack state binding receptors, and the receptors finally regulating the gene expression involving the transcription, nuclear receptors, and those ones. Okay, so the rest of the video left is going is what we are going to do there is we are going to elaborate these receptors, these mechanisms, right? So the rest of the video is going to cover the transducer mechanisms in detail. Okay, now starting with G protein coupled receptors. Now what are they? They link to the effector through one or more GTP activated proteins, which are called G proteins. Okay, so these are basically रिसेप्टर्स. ये रिसेप्टर्स है जो कि जी प्रोटीन से लिंक्ड है. ओके, एंड दे वन सेक. So receptors here is attached with these effectors, which can be enzyme, channel, or carrier protein. Now how the receptor is connected to these? By G proteins. So G proteins here are working or acting as intermediators. Okay, so it is just linking what G proteins are doing here. G proteins, what are they? They are GTP activated proteins. What they are doing here is they are linking the receptors to the effectors. Okay, so receptors we are having जहाँ पे ड्रग्स जाके कंबाइन होने वाली है, एंड इफेक्टर्स वी आर हैविंग जहाँ पे रिस्पॉन्स प्रोड्यूस होने वाला है. ओके, सो जी प्रोटीन आर linking both. They are not directly connected with each other. Okay, so in inactive state, GDP is bound to alpha subunit. Okay, so basically G protein की different subunits होती है, alpha, beta and gamma. So inactive state में क्या होता है? जब ये receptors में drug molecule attached नहीं है, substrate attached नहीं है, तब क्या होता है? Inactive state में GDP bound होता है alpha subunit के साथ. So it is having a subunit called alpha जहाँ पे GDP attached होता है. अब वो जो alpha subunit है, it is having ATPase activity. Now what ATPase does is it hydrolyzes GTP to GDP. Okay, so basically इनएक्टिव स्टेट में जीडीपी क्यों अटैच्ड है अल्फा सब यूनिट से? क्योंकि अल्फा सब यूनिट में एटीपेज एक्टिविटी है, वो जीटीपी को जीडीपी में कन्वर्ट कर दिया, एंड द जीटीपी देन द जीडीपी it remains attached. Okay, now what happens when the receptor is activated? It leads to displacement of GDP by GTP, which either activates or inhibits the effector. Now when the receptor is activated, how it is activated? When the substrate or drug molecule attaches to the receptor, it leads to displacement of GDP by GTP. Okay, which can either activate or inhibit the effector molecule. What are the effector molecules? Different types: enzymes, channels, or carrier proteins. So when the receptor is activated, either an enzyme or channel, they are going to be get activated or inhibited, right? So we are having three major effector pathways for G protein coupled receptors here. Okay, so these are the major effector pathways जिससे जो final biological response है, which is going to be produced by these major pathways. Okay, so the first one is adenyl cyclase CAMP pathway. the second one phospholipase CIP3 DAG pathway, and the third one is channel regulation. So we are not going in depth here in these major pathways. You can read it from the book, right? So the next thing we are having, the second one, what we saw here was that transducer mechanisms were of five types. G protein coupled receptors we saw. Now the second one is ion channel receptors. Okay, so we are going to read this in detail now. Ion channel receptors. So these enclose ion selective channels within their molecule. So see this figure, uh, a receptor is displayed here, right? So this hole is a receptor, and within its molecule it is enclosing a ion selective channel, right, through which ion can pass. Ions, for example, sodium, potassium, calcium, chlorine, it's shown here, right? So these can pass through these channels. Okay, now the agonist binding opens the channel and cause polarization or hyperpolarization or changes in cytosolic ionic composition. Now when the agonist binds, right, when the agonist binds to these receptors, what happens is चैनल में कुछ चेंजेस होंगे. ओके, उससे चैनल ओपन होने वाला है जैसे ही एगोनिस्ट बाइंड करता है, ओके, रिसेप्टर को. व्हाट हैपेंस इज चैनल ओपन हो जाएगा. चैनल में ये तो पोलराइजेशन हो सकता है, हाइपर पोलराइजेशन हो सकता है, साइटोसोल जो है यानी साइटोप्लाज्म जो है सेल के अंदर का, उसकी आयनिक कॉम्पोजिशन चेंज हो सकती है, राइट. Now, agonists directly operate ion channels without second messenger or coupling protein. Okay. Now, in this, what is interesting about this, which differs it from G protein coupled receptor, is what happens here is there is no second messenger or coupling protein involved. Like in G protein receptor, we were saying that a G protein is present which is linking the receptor and the effector, right? Now here there is no intermediate present. The receptor is directly linked to the effector. Okay, so there's no second messenger and agonist can directly operate, yani ki jo drug molecule or the substrate is there, it can directly operate the ion channels. So that is what happens in ion channels receptors. Now the third one, the transmembrane enzyme linked receptors. Now what are they? These receptors utilize mainly by peptide hormones. Okay, so ये receptors पे mainly attach होते हैं as substrates peptide hormones, for example insulin. The enzyme at cytosolic side is generally a protein kinase. Now you see the figure, we are having the cytosol, cytoplasm, which is inside the cell. We are having a membrane and the outside membrane portion. Okay, so in the cytosolic side we are having an enzyme, okay, which is called protein kinase. Generally it is protein kinase. Now when unligaded monomeric state kinase remains inactive. Okay, so when unligaded, what it means, when the substrate is not attached, the kinase remains inactive. Okay, obviously, so it is inactive at that state. And when the hormone binds, the peptide hormone binds, it brings conformational changes which activate the kinase. Okay, now the substrate proteins are then phosphorylated and released to carry forward the cascade of phosphorylation leading to the biological response. Okay, now the substrate proteins जो सबस्ट्रेट अटैच हुआ उसको फॉस्फोराइलेट, ये जो काइनेज प्रोटीन काइनेज एक्टिवेट हुआ है वो क्या करेगा, फॉस्फोराइलेट कर देगा सबस्ट्रेट को. एंड फर्दर फॉस्फोराइलेशन की एक पूरी सीरीज स्टार्ट हो जाएगी जिससे फाइनली हमको जाके मिलेगा बायोलॉजिकल रिस्पॉन्स. ओके, सो दैट इज़ व्हाट transmembrane, that is how transmembrane enzyme linked receptors work. Now the fourth one, transmembrane jack state binding receptors. Now what are they? The agonist induced dimerization. What happens with these receptors? Agonist induced dimerization cause conformational change of receptor which increases affinity for protein kinase. Okay, so here when the agonist or when the agonist what it does to the receptor, it induces dimerization. Okay, so in this figure many receptors are shown. Okay, so when the agonist binds, it induces dimerization और basically conformational change हो जाता है receptor का, जिससे उसकी affinity बढ़ जाती है for some protein kinases which is called JAK. Okay, so JAK is nothing but a protein kinase, right? Now when जब रिसेप्टर की अफिनिटी बढ़ गई, ओके, जैक के लिए, तो व्हाट जैक डज, इट अटैचेस. ओके, एंड देन व्हेन ऑन बाइंडिंग जैक गेट्स एक्टिवेटेड. तो जैक जैसे ही अटैच हुआ फ्रॉम द साइटोसोलिक साइड ऑफ द रिसेप्टर टू द रिसेप्टर, तो वो हो गया एक्टिवेट. और अब वो क्या करेगा, इट फॉस्फोराइलेट्स अ फ्री मूविंग प्रोटीन विच इज कॉल्ड स्टैट. ओके, सो जैक जो कि प्रोटीन काइनेस था, उसकी जैसे ही अफिनिटी बढ़ी रिसेप्टर के लिए, वो जाके अटैच हो गया. देन इट फॉस्फोराइलेट्स अ फ्री मूविंग रिसेप्टर विच इज कॉल्ड फ्री मूविंग प्रोटीन विच इज कॉल्ड स्टैट. ओके, सो स्टैट स्टैंड्स फॉर सिग्नल ट्रांसड्यूसर एंड एक्टिवेटर ऑफ ट्रांसक्रिप्शन. सो जैसे ही स्टैट हुआ फॉस्फोराइलेज, वो भी हो गया एक्टिवेट. उसका काम है ट्रांसक्रिप्शन को एक्टिवेट करना. सो दिस इज देन अगेन गोइंग टू प्रोड्यूस द बायोलॉजिकल रिस्पॉन्स विच इज transcription and all, right? So that is how transmembrane jack state binding receptors work. Now the last one, the last kind of receptors, we are having a receptors regulating gene expression, right? So these are the transcription factors and the nuclear factors. Okay, now intracellular soluble proteins which respond to lipid soluble chemical messengers that penetrate the cell. Okay, so these receptors, these are nothing but intracellular soluble proteins. So these are present intracellularly. The other receptors we saw they were present on the membrane, right? Now these receptors are differently located. These are located intracellularly. These are soluble in the cytoplasm. So they respond to lipid soluble chemical messengers that penetrate the cell. Okay, so we know that lipid soluble drugs, they directly can penetrate the cell. So what they are going to do in the cell, they are going to interact with these receptors. Now when hormone binds to these receptors, conformational change occurs to the receptor and a complex is formed. Okay, now when the hormone binds to these receptors, okay, क्या होगा, हमेशा की तरह कन्फर्मेशनल चेंज होगा. और जैसे ही कन्फर्मेशनल चेंज होगा, हमारे पास बनने वाला है एक कॉम्प्लेक्स. ओके, नाउ व्हाट दिस कॉम्प्लेक्स इज गोइंग टू डू. सो बेसिकली हमने देखा कि ये जो रिसेप्टर्स है वो साइटोप्लाज्म या फिर न्यूक्लियस के अंदर प्रेजेंट होते हैं, राइट? सो दिस कॉम्प्लेक्स attaches to the specific DNA sequences. So we are having DNA sequences in nucleus. So this complex attaches there. Okay, या फिर we are having some target genes, there it attaches, and it facilitates or represses their expression so that specific mRNA is formed. So what this complex is doing, this is getting attached to the DNA or the genes, and it is facilitating or either repressing the expression of some genes. Okay, so that a different mRNA is formed, right? So when different mRNA is formed, obviously the biological function, it is going to get altered, right? So for example, all steroidal hormones function in this manner. Okay, so all steroidal hormones are functioning by receptors regulating gene expression. Okay, then the last topic we are having here is regulation of receptors, right? So again, it is important that how to regulate these receptors, how they are regulated, right? Now the receptors, they exist in dynamic state. Okay, so what is dynamic? The it is प्रेजेंट इन एक्टिव स्टेट, मतलब पूरे टाइम कुछ ना कुछ चेंजेस होते रहते हैं, राइट? कुछ ना कुछ मूवमेंट, कुछ ना कुछ something is happening there. So it is not static, it is dynamic. Now prolonged deprivation of agonist results in super sensitivity of the receptor. Okay, so what is interesting over here is that जब receptor से जो बाइंड होने वाला सबस्ट्रेट है या जो अगोनिस्ट है, जैसे उसका प्रोलॉन्ग बहुत टाइम तक अगर नहीं प्रेजेंट है एगोनिस्ट और वो रिसेप्टर खाली पड़ा है, तो उससे सुपर सेंसिटिविटी हो सकती है रिसेप्टर की, मतलब रिसेप्टर हाईली सेंसिटिव हो जाएगा, राइट। तो अगर आपने ड्रग दी तो उसका एक्शन बहुत ही ज़्यादा आने वाला है। ओके, नाउ continued similarly continued intense receptor stimulation causes desensitization due to masking of receptor first, then the second one decreased synthesis or increased destruction of receptor. Now what happened here is, अगर आप continuously वाइस वर्सा हमने देखा कि हम डेप्राइवेशन हो जाए या फिर आप कॉन्टिन्यूसली देते जा रहे हो द अगोनिस्ट, व्हाट विल हैपन इज डीसेंसिटाइज हो जाएगा हमारा रिसेप्टर। सो डीसेंसिटाइज हो जाएगा मतलब लेस सेंसिटिव हो जाएगा टुवर्ड्स दी substrate, right? Now how this desensitization is occurring? Okay, so it occurs by either masking of the receptor or by decreased synthesis or increased destruction. तो receptor पे कुछ molecules आके उसको mask कर देंगे जिससे agonist attach ना कर सके, right? And synthesis ही कम हो जाएगा receptors का या फिर destruction ज़्यादा हो जाएगा receptors का. Now we'll see the last topic, functions of receptors. Now the functions. The first one, propagate extracellular signals inside the cell when molecular species carrying the signal cannot itself penetrate the membrane. Okay, so we are having some signals extracellularly, we are having some molecules which are carrying those signals, and those molecules itself, they are not able to penetrate the membrane. So what will they do? They will just pass on the signal to these receptors which are going to propagate the signal inside the cell so that the response can be generated out of it. Okay, now the second one, amplify and integrate the signal. So what they are going to do is they will amplify, will increase the intensity of the signal, okay, so that a particular biological response again is generated. Now the third one, they adapt to short term and long term changes which to maintain homeostasis. So again, every cell, what is special about a cell? So cell is able to maintain homeostasis inside its environment, right, in cytoplasm. So how homeostasis is maintained? It is maintained by short term and long term changes. Okay, so that is how this dynamic receptors they are working, right? So for a short recap, what we saw in this video. So in this video we saw firstly the action effect sequence, what is the difference between drug action and drug effect, right? And then we saw what is the structure of receptor, what are the domains it's having, then the transducer mechanism, what are the different types of it, what are it, right, what is it, and the different types we have seen of transducer mechanisms was first one Gprotein coupled receptor, right, and then the second one ion channel receptors, the third one transmembrane enzyme linked receptors, then the fourth one transmembrane jack state binding receptors, then the fifth one receptor which is regulating the gene expression, right. Then the last topic we saw was regulation of receptors and the function of receptors. Okay guys, that's it. If you found this video helpful, like, share and subscribe, and keep studying, see my videos regularly, and yeah, I'll take leave. Bye bye.